Hi, my name is Sam from Ghana. This year I'm presenting something on the impact of blood collection tubes, that's Vacutainers, on activation of T cells. So blood specimen is very important in biomedical research. So with a little amount of blood, you could actually perform diagnostics. You could determine whether someone is infected, is diseased, or not. You could also use it to monitor treatments and interventions. You could use it for your immunology studies, that's determine the immune response to a particular disease or infection. You could also use it or employ it in your genomics, transcriptomics, and other omics experiments. So the integrity of the specimen is critical, and then the choice of a Vacutainer is important in any blood-related experiment. Today, there are lots of Vacutainers on the market, and you might want to employ either of these depending on the aims and objectives of your study. So for this study, we were trying to determine the impact of time on the frequency and activation of CD4, CD8 T cells stored in EDTA tubes. We compared the results for that stored in heparin tubes. So what we did was to gather a bunch of students, 9 in all, and then we took 5 ml venous blood twice from each participant. So one part of the blood was put into the EDTA tube, and then the other part was put into the heparin tube. And what happened is that we divided each sample into two. So the half was processed immediately within an hour, and the second half was processed after 4 hours. So we performed whole blood staining, and then we performed our flow cytometry analysis. We used these antibodies in our study. So CD3, CD4, CD8, and CD69. Remember, CD69 is an activation marker and helps us to really differentiate between activated T cells and those that are not. So this is something on the study participants. As you could see, they are quite younger, 18 to 23. A lot of them fell in that group. So this is something on the frequency of CD4 T cells in both EDTA and heparin tubes processed within an hour and at 4 hours. So the first figure is something on the frequency of CD4 T cells from EDTA tubes. So this T1 and then T4, you could see there is no significant difference between the two cell populations. Now this is something on T1 and T4. So these are CD4 T cells, the percentage or the proportion of CD4 T cells from heparin tubes processed immediately and processed after 4 hours. There was also no significant difference between the frequency of the two cell populations. So we also tried to determine the frequency of CD8 T cells from EDTA and then heparin tubes. So you could see that at T1 and T4, there was no significant difference of the frequency of CD8 T cells. So we also determined whether there was we also compared CD8 from heparin tubes processed within an hour and then processed at 4 hours, and there was also no significant difference. So basically, neither EDTA or heparin tubes affect the frequency of CD8 T cells in your immune assays. So this is a summary of our expression experiment. Um, so the first item, all right, sorry. So before I move on to the table, I just want to explain this to you. So in our amino acids, usually we expect um, a higher proportion of CD4s to CD8. So I just want to give you this as a background information before moving on to the table. So the first item on the table, we compared the proportion of CD4 T cells from heparin tubes processed at T1 and then time 4 hours. So you could see that there was no significant difference. We showed this in the figure previously. So the second item on the table is that of CD8 T cells. So the frequency of CD8 T cells in EDTA and then in heparin tubes. You could also see there was no real significant difference between these two guys. Then again, we used the student t-test to compare to see whether there were any difference between CD4 and CD8 T cells, the frequency of CD4 and CD8 T cells at time 1. So basically, these are from EDTA tubes. And you saw that the proportion of CD4s were quite higher than the proportion of CD8 T cells. So remember, I explained that in our sample, in our experiment, in our amino acids, normally the proportion of CD4s are quite higher than CD8. So this is something which is expected. I mean, this is an expected result, basically. Yeah. So we also did the same thing with samples from the EDTA tubes processed at 4 hours. So you could see CD4s were quite higher than CD8 T cells. That was significant. So still remember, CD4s are usually more than CD8 T cells. So this is expected also. And then we compare the proportion of CD4s and CD8s. All right, in heparin tubes, that's significant. And then so we compare CD4 and CD8 from heparin tubes at time 4 hours. That's also significant. Then again, we also checked the activation of the various T cells from the EDTA and heparin tubes. So this is something on CD4 T cells. So these activated CD4 T cells from EDTA tubes. So you could see that the proportion of activated CD4 T cells were quite higher from samples processed after 4 hours. They were quite lower when you process them immediately. Now there was a significant difference between these two cell populations. So in EDTA tubes, the proportion of activated T cells are quite higher when the samples are left for some time before processing. That's the conclusion we can draw from this graph. On the other hand, this talks about the proportion of activated CD4 T cells from heparin tubes. So you could see these are samples processed after 4 hours, and then these are samples processed immediately. There was no real significant difference between these two guys. So basically, you can't really draw the conclusion that samples left after collection, or samples left maybe after 4 hours. There was any real significant difference with samples processed immediately and samples left after 4 hours. Sorry. So this is also a summary of the T cell activation of samples processed at 4 hours and then samples processed immediately So you could see that there was a significant difference between um, activated CD4 T cells and EDTA Ts. I think we showed that on the figure. But then when we come to the heparin tubes, it looks like the cells are quite stable. The proportion of cells are quite stable. So the 0.600, 0.622, no real significant change within the cell population. But I mean, if you look at the EDTA tubes, the 0.38 and the 0.7, man, that's quite high. So that's why it was flagged. So we could also check for this. So we used, I think, the Mann Whitney to actually determine whether there was any real significance between the proportion of activated CD4 T cells and then CD8 T cells. And yeah, there was a significant difference between the two cell population. But then remember that I told you from the previous slides that in our immunoassays, in normal immunoassays, we usually expect CD4 T cells to be of a higher proportion than CD8 T cells. Yeah, so this is quite expected. So our conclusion is that activation of CD4 T cells, CD4s and CD8 T cells in EDTA tubes differed significantly at time less than 1, but not at 4 hours. So heparin is a more stable anticoagulant for immunological analysis compared to EDTA. So recommendations, we might want to try other specimen tubes. You might want to try using different specimens other than blood, or try different experimental approach or experimental setups. So these guys really played a major role in the development of the study.